All right, everybody. Welcome to today's webinar put on by Elucidata. My name is John Conway from 2015 Visioneers. Today's title is From Raw Data to Curated Resource, Navigating Complexities of Single-Cell Curation. Today's agenda, we'll give a brief industry perspective. I'll do speaker introductions, and then Elucidata will give their presentation, followed by Q&A. So kind of our rules of engagement: put questions and or comments in the chat or QA box. General questions will get moderated after the talk. Everyone will have access to the recording afterwards. So a brief industry perspective, 2015 Visioneers. We'll have some definitions, some data science, and data curation. So in our opinion, FAIR principles and model quality data are imperative for the future of drug and therapy discovery. Too many times, organizations are mired in 80% data wrangling at worst. It's very inefficient to find data, to use your data in research. It takes heavy lifts many times. So companies like Elucidata are cleaning up data. They're creating fair data environments, and they're providing tools for that deeper analysis, deeper insights. So really, that findable, accessible, interoperable, reusable with the capital R is critical. And in companies like Elucidata, producing model quality data, it's imperative. So having high data integrity, highly contextualized data, confidence in your data, and then applicability. These are just really important things and areas that you want to take into consideration when producing these data environments. Data science is a broad approach to a lot of this, and doing this in a methodical way, but also taking into account what's needed to produce model quality data and curate data, et cetera. So you have to have a strong understanding in computer science, in math and statistics. You have to understand the domain you're in and the intricacies, the details. And when this all comes together, you are working in software, data analysis, you're doing advanced algorithms and machine learning. This is where data science all comes together, and it does take all these different disciplines to achieve that. Then lastly, data curation is critical. When you can curate data from around the world and bring it into your research and your work is a great ask and a great lift. But you really need a strategy around this. You have to have a strong strategy in place. You have to understand the data that you're dealing with. So it takes data assessments to figure out the good and bad parts of the data that you're working with. You have to understand how and what to contextualize, the annotation that you bring into this, how you're going to extract it and ingest it, and ultimately make it a fair data environment. So there is a process to this, and this is one of the great things that Elucidata brings to the industry. So Risha is a curation solutions manager at Elucidata with more than 10 years experience developing algorithms and generating insights for drug discovery. She's involved in understanding customer data curation requirements and translating them into scientific and technical plans that help customers achieve their research goals. She also helps in building scalable and machine learning-based solutions for public and proprietary data curation, custom automated workflows, and data analysis. Shruti is a leading bioinformatics engineer at Elucidata with more than 8 years experience developing algorithms and generating insights for precision medicine. She manages the ETL, extract, transform, and load of all the biomedical data that Elucidata's cloud platform, Poly, harbors. She has been influential in ensuring all of Poly's data adheres to FAIR principles and is curated as per client requirements. So with that, I'm going to hand it over to Elucidata. Welcome.
Thanks, John.
Sure.
Right. So thank you, John, for setting the stage for us and, recapitulating of what we do here. So particularly today, what we want to talk about is our experiences of dealing with curation of single cell data sets. And I'm saying this like these are our experiences, but I think it's more about our struggles and our finally our ways to come around to understand how we can extract information and curate that information for single cell data sets and make that available to the use to our customers, ready for exploration and so on. Yeah, next slide. So, I mean, before I jump into anything related to single cell, the first and key thing is that with the advancements or with the technology that has come, it's been termed as one of the most promising the new age technologies. What it promises us is now that we are able to even ask biological questions, which are very cell-specific, cell type changes related. Previously, when we were asking questions, it was a very we would compare very heterogeneous systems and we would try to find some clues and some significance. And now with single cell, we are able to go to a much more granular level of single cell and answer questions. So like key differences, like we were looking at immune profiling, but we were always looking at the whole the tissue changes as such and looking at looking at genes which would differentiate. But now we have the capability of even going to a much granular level and see how the cellular heterogeneity is changing itself. If we were to compare two different cohorts with disease versus normal cells, normal patients, then how would these also affect in terms of the change in the environment and how does that go on? It's another key thing that we have that we single cell has enabled us to do is to identify changes in disease conditions which are not to say with all of these possibilities. What's coming clear is that the better we are at first identifying these cells and the better we are in understanding what is in the normal system versus how it changes for a disease system is extremely crucial. And it's very important for us in cell type identification.
But Richa, like I have lately seen experiments are becoming more and more complex and more large scale. And cell annotation, like as we have gone through a lot of data sets, like cell annotation is not that straightforward. Even the information that we get with this with the publications are variable in complexity and context, right?
Right. Absolutely right. Shruti, that's exactly what we faced when we started, curating data sets for different customers. Initially, it was I mean, it kind of felt like, oh, we will get dependent, and it requires a expert-driven process as well. So, I mean, initially, also again, like when we started isolating cells and we were, we put them in reaction chambers, we kind of label them in barcodes. It seemed like yes, it would be easier to identify these cell types, but more often than not, it is not very easy. So what we observed and what I want to share with the audience today is that there is a spectrum of information which is available to us with the publications that help us in cell type annotation. So the first and the easiest way, these are my favorite authors who, when they make a data set available, they are very generous and they make all of the available metadata and cell type annotations that they have used in the experiments with the data sets itself. As the first case at hand is that there would be metadata files which would just simply give us the cell ID and also map them to the corresponding cell type that each of the cell is really identifying with. And such kind of such information, when available, is easy to replicate and add to our curations data sets that we have. They may have some other, sometimes there would be an indirect way of finding information, but nonetheless they make it available in terms of the tables and figures. So as you can see, the figure on the right, the authors made available the clusters with the data set as cell metadata. And then if you look at the publications, you can clearly, unambiguously map their cluster IDs with the identified cell types. And this makes our job, this makes at least the accuracy of or replicating this author cell types much more simpler in this case. What is key to understand is that at this level, that most of whatever information is available is unambiguously getting mapped on. So many times when we get data set, they might be filtered, and the metadata that we get is maybe of all the cells that are there. So one has to be a little careful in mapping these, but more or less we can unambiguously map the cell ID with the cell type information, and this becomes the easiest way of us to annotating cells, so on. Next slide. Next. Right. So then the level of complexity that comes up with is that now the authors may not have made this cell type information available, but they do have ways of providing that information through the marker gene list. So in this cases, what the authors generally give is it's not an easy way, but in general, they would have to us that which have been studied the scenes for each of problem that at which we want. I mean, there will be study. They could define very broad cell types, like the neuronal cells or epithelial cell or immune cells. And like in like you see in the first case, that these were all these were not uninflamed and inflamed tissue from Crohn's disease patients, and what they wanted to study was at a much high level of what are the B cells, plasma cells, and the stromal cells. And then for each of them, they had provided the markers that they had used to identify the cells that they have isolated and studied. But then you see the case in the second one where on the right, that they wanted not just to study at a much granular level. So when they start so basically in the profile that they are trying to profile of cells they're trying to do, they would also then further subdivided based on the marker that are present. Suppose they want to measure, say, immune cells. The broader terms would be the T cells, B cells, and so on. But if you can further go ahead with the markers that are available, you can also have the granularity of putting cytotoxic T cells and T helper cells with CD8 markers and CD4 markers respectively in these cases.
Right. But Richa, then like when we talk about these levels of identities, right, there happens a problem with generating the consensus of cell identity, and many a times we don't actually come to the consensus part of it, right?
Right. So this is Shruti, this is still an open problem, I would say, that one of the well-known reasons of why there's a difference in the cell type consensus over what we call cell type identifications is mainly what the authors are trying to study. It's if they are trying to study, say, the cell fate and how the cells are evolving from one state to another, then the granularity is much more expected. But if they're trying to understand just the heterogeneity between two different cohorts or two different conditions, then the granularity might be lesser for them. So it's kind of dependent on what the authors want to do, and then exactly those markers are what they kind of identify. And the other is also that, if we look at, like, we do now have some exhaustive, databases to identify cell type markers, and canonical ones that we can use. They're pretty exhaustive, but still there is ongoing efforts with atlases and different conditions with different atlases to come up with novel cell types and then their markers that could be used. So that's another thing that we do not at this point have, like, a comprehensive, and we're still discovering new cell types. So that's one of another reasons why we are not having a consensus of which we want to do. But yeah, I mean, I think we will be discussing this also in your, this piece of information is useful in even annotating when you have these marker gene lists.
Right.
So quickly moved on to my worst cases, which is the next slide. So there were some authors who at least provide either anamorphous management or at least provide some clues, like cell type markers. But then we have often come across data sets where there is no information available. And no information available is essentially is that they might not have used the entire data set to be analyzed by them. They might just use a subset of this data, and the remaining all cells are unannotated, not provide any markers for it. So those could be, some very important cases, or sometimes, the UMAP or the TSNEs are not well, and figures are not very well annotated for us to, derive information. So we definitely even find such, some of the publications which they have no information to go on forward with. So with that, like, if we go to the next slide. So what we see is what I'm trying to summarize here is that there is a spectrum of information that's available, and with this information that's available is how we would then work towards getting the more accurate, and this would define our effort to identify cells and annotate them with the right cell type. What is critical to understand is, like, as we know that there is a difference in how, firstly, in the definition itself of the cell type, how authors would use. The other is that they would also use different terminologies. In our when we curate data sets, so it's not just about collecting all of the information that we're consolidating it, but also once we are able to unambiguously annotate cells, we also would harmonize them with our cell-based ontologies. And yeah, these are some of the very critical aspects that we consider while curating data sets for our customers. So, Shruti, I think I would love to, if you could just talk in details about the curation process and the flow that we do for cell annotations.
Yeah, definitely. I'm always super excited talking about it, because every time I talk, it's a learning experience for me. So broadly, the process for single cell annotation, and it's quite of gold standard, is like we do a thing of pre-processing, then we do unsupervised clusterings, then we try to annotate the cells, and that's where we get the cell type annotations with the markers. So to talk about each of the cells data sets or steps in detail, is like we at Poly meticulously curate all the data that is there on Poly. And it's not usually a straightforward process, because various studies come with dynamic data formats like MTX, CSVs, Loom files. Even the processing steps are not common, like where some data sets have raw counts, some come without raw counts directly from the FASTQ files. And even the metadata information that is available, right, which would help us in cluster annotations, is of very degrees. So over the time span we have been curating data, we have generalized the curation process to an extent that each study at least has a backbone of what processing we'll be doing. But each data set is a standalone in itself, with varying degrees of tweaks required to make that data comparable with the other curations and annotations that we have on the platform. So firstly, the data is available either in raw or normalized counts. And as I said, few studies might not have the counts, but FASTQ files. few of my favorite authors, as Richa mentioned, right? They give everything normalized data, QC clean data, and steps are already done. So the only thing is like we just need to massage it into the format and add the cell type annotations, and the data is ready to use. But that's kind of an easier case. So let's talk about a more detailed case where we start from raw counts, where we do not have any other additional information, and let's try to decode that process, which typically begins from author provided raw counts data which we have. So the first thing that we do is we come to the pre-processing, which is some basic QC for the data, removing out cells fewer than 2000 genes, and then removing genes which are expressed for fewer than T cells. This just helps in eliminating noise or locality data. Next thing that we do is some additional QC analysis by looking at other matrices for total gene counts and mitochondrial content. Again, we remove the noise from the data by removing out the cells that are having high total counts as compared to the rest of the data, which kind of signifies they might be doublets. Then we remove the cells that are having high percentage of mitochondrial uh, genes content, which might indicate that these are non-viable cells. Then we apply, generally try to apply the parameters that authors have mentioned in that literature, so that we do not divert much from those parameters with the goal of replicating the analysis published by them. So the reason for doing this, as Richa mentioned, right, we there is a perspective with which a author has used a data set and analyzed them. So we just do not want to divert much from that in the process of actually standardizing the data sets. Okay, so that's the pre-processing steps or the quality control step. One caveat is like if we filter out too many cells, right, we might lose rare cell populations, and if we are too lenient, we might just add noise, giving us hard time to actually curate the cell type. So it's kind of a subjective balance that we need to maintain when we are actually doing our pre-processing step. Then the final thing that we do is basically we do a total count normalizations for about 10k cells, 10k reads per cells, and then which basically reduces the variation arising from the sequencing data. Then the log data is log transformed, scaled, and then we annotate the highly variable genes, which are mostly the representative markers of meaningful biological variations that is available in the samples. Few times we do observe batch effects with respect to the processing or the protocols being done in different batches. If we encounter those kinds of data set, we try to remove the batch effects using the metadata available with the respect to experimental design or sample handling. Once this is done, like the next step that we come on is like unsupervised clustering, for which an important step is dimensionality reduction, which we reduce the data complexity and visualization, which the gold standard is principal component analysis, using keeping only the highly variable genes that we have found in our earlier step. We generally take the threshold of the principal component PCs to be the flattened elbow if the information is not available around in the publications. If it is available, we just try to stick to that information. Then we compute the neighborhood graphs of cells, PCA representations on data matrix, and derive the embeddings for neighborhood communities using the UMAP or t-SNE plots. Finally, once we have the neighborhood graphs, we do laden clustering method, which is kind of a bit iterative to and fro method with different resolutions to get the clusters or the number of clusters which are in sync with the author published cluster graph. The next crucial step is basically the cell type annotations using the markers mentioned by authors. If some datasets we do not have the marker information associated with the cell types, that is where we actually resort to canonical markers from publicly available studies or databases, and we use the expression patterns of those markers to annotate the cell types. We visualize the data using the individual markers first, or the set of markers when we don't have the markers available, and then we annotate the cell type of interest. Additionally, we also generate the clusters marker by cell type plot to get a holistic view of whether there is no overlap between the markers and the cell type clusters that I'm trying to annotate. Additionally, we use some quantitative methods for cluster identification using automated algorithm, but that algorithm does need marker inputs from the user. The reason we need to do these ensemble of methods is to actually get out the accurate cluster annotations, because individual methods sometimes not work in all of the data sets. Once we have this cell cluster annotations, right, that's the hard part done. We normalize the cell type label using the cell ontologies. Fewer times it so happens like the cluster labels that are annotated are not available as part of ontological databases. In such a case, we try to put a one layer above or a global cluster label if it's not confusing with the originally available clusters. For this whole process, currently we use Scanfy because that's a go-to tool for majority of analysis that we have seen in Cell and Nature papers, and it also integrates seamlessly with our tool and works pretty well with the larger data sets. Nonetheless, the final curated data sets that we have on our platform, right, are available in Curat compatible format. So Curat is also an equally widely used tool in single cell data analysis. So that is pretty much the process that we follow for our cell type annotations.
Yeah, Shruti, I mean, this sounds like a very elaborate. I mean, we of course have to do a lot of QC steps, and before we can even start cell annotations, we have like quite a good process in place. But does this mean like it's this straightforward for all the data sets, whichever have marker genes available? Is this a straightforward process? Can anybody help us do that?
No. So, I mean, straightforward is probably not the word that can go with cell type annotations rather. So there are like diverse scenarios, and most of the cases we tend to diverge here and there from the standard workflow with varying degrees of complexity. So I have added a couple of examples, like not exhaustive, but just two important ones, which are kind of very much recurring in whatever data sets that we see. So first one is basically the ambiguity in the cell type annotation. So what I mean by this is like the cell markers, right? The cell markers are not clearly demarcating the cell types. So let's look at this example, which is about the leukemia stem cell. This is quite an extensive analysis the authors have performed, no doubt about it. But if you see the marker expressions versus the clusters versus the cell types, right, in the top left figure, it's very evident that the annotation is not going to be straightforward. So what we generally do is like we resort to a stepwise process. So what we do first we annotate the cells and the clusters that are free of ambiguity, like the GMPs and the monocytes that we see here in the top middle figure. And then we assign placeholders for the rest of the cells, which are generally the cell types with a one level up or a global or a broader classification level. And then then what we do is, either if the markers for those niche cell types are given by the authors, we try to do expression analysis for those individual set of markers, or if they are not available, we generally go to the canonical markers, and using those markers, right, finer marker expression profiling, we add the cluster map over there. There is another example which is a bit more trickier and is kind of more consuming problem for us, wherein we do not have a marker information or cell type annotation is available, which basically I'll talk into the next slide. So this example that we see, right, the cell types are differentiated into clusters, and the authors have given the nomenclatures for those clusters, but the associated marker information is not available. Sometimes even the nomenclature for cell types is not available. That is an altogether different level of question that we're still trying to solve. But when we have these marker cluster information given, We acquire the canonical markers for these cell types to annotate the cluster from the publicly available databases. Now, this is again not a straightforward as we see here, like we just hand in some markers. We need to fine tune the clustering parameters to a couple of permutation and combinations of the markers that are expressed canonically for the required cell types, unless and until we get not very clearly defined, but a bit well defined cluster annotations as we see here in the middle graph. And not so rarely we actually are unable to get the finer cell types after considerable analytical efforts, after considerable fine tuning and permutation and combination. In this cases where the direct marker information is not available, what we do is we resort to annotate the broader cell types rather than sticking to curate or not curate the finer details, right? So one line of thought here is, which is also applicable to the most of the data sets that we have, is splitting the data set too much from the beginning itself, right? It's not always going to be a wise case. So too many clusters can make our annotations more challenging, especially when we are dealing with heterogeneous data sets. So a useful trick that people can use or we can use is like subclustering. Users can find clusters, annotate it at a moderate level or a global level, and then take out that smaller clusters and then try to treat it as a new data set and do another set of analysis to find some interesting cell subtypes, which can be explored more easily given the rest of the cells with noise are removed. So we have done these kinds of treatments to few data sets which actually had some interesting insights into finer clusters than global clusters. But the bottom line is that like curations are done keeping the publication or the literature in mind and in sync. The processed and the normalized data set is retained in the data set information. So if someone actually wants to find some novel insights from the data sets, uh, which is not a part of the original data and is very divergent from the analysis that the author has done. So we do keep the raw data with just the bare minimum QC processes done on it. So here actually I wanted to ask you, Richa, there I have come across a lot of situations and you have been interacting with a lot of customers, right? Considering this scenario wherein there is a data set of interest, but the analysis that the user wants to perform is not restrictive to the original analysis, right? So the user might not want to actually have the cell types that are available in the paper, and he wants to do some novel innovative analysis. Besides cell types, which are any other important annotations they look to find the data set related to them?
Right. So I'll answer this in like two parts. The first one relating to, that do customers want raw data as well, or are they, or do they want author annotations and so on? But, public data is essentially for reusability. And I have had many customers who would want to first understand what the authors, like what the publications have done generally. And you very rightly point out like this is a very widely used technique where you first do the clustering on a broader level, and then you would do when you're unambiguously able to annotate broader cell types, you can then further go into a granular level. So I've had many customers who are looking for that at different levels of clustering as well. So whatever is provided from the authors, then moving forward to what they can also do further with the data. And in that sense, providing them with the raw data and the process come both together with these annotations or the publications is often very useful, and a very common ask. But as the second part of the question that you want to understand is this what they all like? Do they know what data sets they want and so on? And can I also help them with finding relevant data sets related to their biological question? So the answer to that is, of course, yes. The more the metadata, the richer the metadata is better for any type of them for repurposing those data sets for their own needs. And here again, there have been, very different type of challenges itself. So as you could like, so over the time, what we figured is that for single cell specifically, since there are no standard ways, there is a lot of heterogeneity in which the data is even deposited at different sources. And within a source itself, if the data set is deposited, even then we would find a lot of heterogeneity. And what I mean by heterogeneity is like there's a
Right.
breadth of files in which you have to search and get your information from. And what we want to do is like we want to collect all of the original metadata that we have, and enrich that and standardize that in a way that it would help our users to find what they're looking for. Just giving examples of what the breadth of metadata, like the favorite authors would always provide a file which would be the Excel metadata type, and that's the most useful one. But many times that these are patient clinical information, so they would have sample level and patient level information, which may not always be part of like the cell metadata file, or you probably have to derive that from the publication itself, from the figures of the publication, or supplementary files as well. And then
Right.
these need to be mapped to the metadata to the data we have, which often is not, which is something that we have not like found as a very straightforward or like easy to do. And what we have tried to do at Elucidata is try to make that a semi-automated method. One very interesting example we saw sometimes where like there is sample description which encompasses a lot of information that may not be available for sample, but you split that data itself, that would we would be able to get more information. There are examples where the mapping is not very easy between this cell metadata that we have found versus the sample. There would be differences in how they define the sample ID. So that needs a little digging, that needs a little bit of understanding the publication itself, and finding and correcting those. And we often even use information related to the cluster numbers, and how the counts and the number of cells in a given cell type. So those things to unambiguously map the sample information to to our metadata as well. But yeah, making all of this, trying to extract all information from different samples and descriptions, and unambiguously map all of the scattered information, trying to collect it, make it consistent, and that definitely enriches
So
the data.
so how do you actually try to make it available for the customers, right? Because like as you said, right, the representations of author for diseases or cell types might be different. So
Right.
are there any standard procedures that you apply throughout the data that's there on the cloud platform?
Yes. So yeah, I mean, simple answer, yes, but the work behind it is a lot. So clearly, like first is metadata richness. So we have all of the original metadata that exists with this. But like you said, like everyone has a different way of, different way of even calling the simplest of things. So if like showing you the example here, like we could call COVID-19, or we could call it coronavirus disease, or we can call it SARS infection, all of which we know are actually indicating to the same COVID-19 disease. So such type of information needs to be harmonized, and one of the ways that we have identified is to use ontological terms. But again, this is something that we wanted to do at scale, and at Elucidata, what we try to achieve here is that we have made different models that would that would first identify these terms from the text or the sample information and so on, and then add the ontological term for it, which would be consistent across data sets, and which would enable us to further, when we're searching, then we have a consistent way to query it. And it's not just like we're not using multiple keywords to get to the same data sets. We could just have one keyword, which is the ontological term, and be able to find a data set much faster and quicker on the platform. So just like very briefly, and like what are the different as like we in-house have curated to say about like more than 1,800 data sets from they might be split from different publications, and we have curated multiple cells, about more than around 800 cell types and so on. But all of these are powered by our polybird models. Essentially, what we try to do, if you look at the source, you have missing information, and you do not have them represented in standard terms. So, but the data which is curated on poly is what we call ML ready, because now these all of the missing information has been added onto it. Plus, also we have replaced them. We have added this ontological terms that are. And adding ontologies, like I would like try to emphasize here, it's not just like powering our searches, because it's not just like we're harmonizing the term, but it is also it's also enabling our customers to find comparable data sets. Most often they're not looking for one data set, but they're looking for like a couple of data sets on which they can validate. So they would. It also helps them in finding those comparable data. And another key usage of having ontology is that when you make have these standardized terms, then the downstream workflows are often very easy to implement. So it becomes an automated way for us to plug into downstream workflows, which could use these curated fields standardized to our sense. And one more advantage that I can think of is that when you have ontologies, you can make much more complex queries. You are not using multiple keywords, and you're able to make more complex queries with much simpler, shorter forms, and so on. And it also enables us to then helps our customers to make new discoveries and so on.
Yeah, it's amazing. Like we have cell type annotations ontologically cleaned up, and the metadata annotations, and so this actually triggers me to ask a question. So can I actually find a data sets which is about immune profiling, maybe monocytes or those kinds of cells from an autoimmune disease? Let's talk about lupus. Is that a easily doable thing on the platform?
Most definitely. Maybe I can quickly share my screen and show. Just give me a second. So is my screen visible?
Yeah.
Right. So yeah, I mean, so this is our Omics Atlas, and so this is where we have consolidated, we have curated, and we have added all of our single cell data sets that we have curated so far. And what we make available on the platform is whatever is ontologically enriched is something that we can, very simply use as filtering techniques or even querying techniques. So simplest way is, of course, you have a search bar. You can type what you want to, say search lupus and, find what you want to. But because of the ontologies that we are following and we are adding more and more each day, so this platform is also very flexible to incorporate those new changes for them. We are able to then use each of those as say filter things. So suppose that I'm looking for, like you said, you want to look for disease which is lupus, so we can search for lupus in here as a disease term. And yeah, it shows that we have about like 16 data sets which match the criteria of like this disease being associated with lupus, or some patients would have some clinical symptoms as well with lupus as well. And in this, if we want to search for such different cell types, then we also have we have these additional information. You can also choose cell chemistry of the assay types that was used. But yeah, I mean, I would say just say CD8, 48, T helper cells, regulatory cells, so we have 5 different matches here in our data set. And what the platform enables us is like very quickly have a look at these data sets, or understand like what these what is the metadata that's there, what type of experiment that has been included. Within this, you can see a description. These are different tags that we have added to make this data set more queryable, and based on what the standard fields that we have curated for these. And if you look at the data set metadata overview, this kind of gives us a clear picture of what whether this data set has healthy or control samples or not. So if you can see like disease type here is lupus versus normal, and you can see all of this difference, different cell type information, cell types that have been come up. You can see that this is a large data set of more than 100 200 key cells in this. And this also, like, I mean, the platform is like pretty flexible. You can see different pie charts to visualize your metadata as well. It's not going through those tedious tables trying to understand what all has been added. And another thing that we one of the of our customers really appreciate is like, can we quickly even analyze this, or like have a look at what is the entire available metadata from the source as well. So we have the options of analyze, and what we have done is we have linked this to Open by Cell by Gene. Is one of the most widely used interactive tool for exploring single cell data sets. And what I quickly wanted to show is that once we have this data, we can choose a workspace in which we want to open, and then we can explore this data. So yeah, in the interest of time, we have just added explored this data already in Cell by Gene, and like for new people who have not used Cell by Gene, maybe a slight promotion here that whatever categorical metadata information that we have is available on our left panel, when it's easily whatever type of visualization you want GSNE or you want UMAP, different types you can embedding choice you can use, and then you can further populate these like try to see how these categorical information kind of map onto this. And especially the fields that you see curated ones are the ones that we have curated and added ontological terms for these, which makes it comparable across different data sets and so on. Yeah, I think that's what I wanted to quickly show you here.
Rita, there was a question in Q&A where the attendee wanted to just see the UMAP and TSNE. So if you can just show it on the same data set.
Sure. Sorry. My info panels come here. So yes, so if I just toggle between the different PCA or TSNE UMAP for this data set, this is how it would look.
Cool, thanks.
Cool. Be open to questions.
Okay. Let's go into Q&A. Let's see. First one, is there a difference between what you were showing? I think it was on the first couple slides in single cell R-seq data. So I think there's a question around how much R-seq overlap or on what you're doing, like the difference from just getting the the raw data, etc.
I'm not sure which slide was this on. Something that I was presenting over.
Yeah. Let me clarify this with Tanmoy. I'll tell you what. If he's still here, Tanmoy. So Tanmoy, I promoted you. You can ask the question if you want to.
Yes, ma'am. I was asking. Just a minute. Ma'am, I was just asking that whether the total analysis, and is there a difference between the single cell RNA-seq data that we use? What is the difference between this and single cell RNA data sequencing?
So these are also single cell RNA sequencing data itself. I mean, are you talking about bulk
Yeah,
RNA sequencing?
yeah, bulk
Okay.
single, yeah, between
Right.
this one and that one.
Oh,
All right. So yeah, I mean that's so basically, I mean, historically when RNA sequencing techniques were, which came about, what we were sequencing at that point was the entire tissue sample. So suppose you have a tumor, a cancer patient with some tumor growth, you would take the entire tumor tissue for that, and then you would be trying to, and then you would be, homogenizing it and trying to measure the, RNA count. So essentially, in that case, what you get is like an average of what happens in your entire tissue. But since we understand that a tissue is made up of very different type of cells, and each cell, there's a lot of cell-cell communication. So not every cell is reacting to when you have a cancer type or something. So that information actually, difference actually gets lost. At single cell level, what you can really do is understand what is happening in just that type of cell in its own. And it's what we have seen in the very classical papers that have come up, is that it's not just about changes in gene expression. Sometimes it's even the composition of the cell type that changes with different conditions, and that information is completely lost when you're looking at the whole tissue level, and you're not looking at some, at granular level for the cells and so on.
Yes, ma'am. One more question that
Mhm.
I have used RNA16S, RNA6 data for analyzing that metagenomics data. And there I was trying to find out the correlation between the microorganisms in the gut and what kind of response that they have on the immune system, and can they cause any autoimmune diseases. But it was a total failure. I couldn't do that because the metadata was not giving me the pipelines that were using. That was not giving me that kind of data. So instead, I end up discovering this whole new series of unknown species and all, but the main objective was not achieved. It became a totally different what was my aim and what the conclusion came was totally different. So, ma'am, with the poly, can we identify the particular organisms that can lead to this kind of can we identify, so it can be
Right.
identified which kind of autoimmune diseases can be caused by this kind of microbes or something like that?
Right. So yeah, currently at Poly, the two data types that we help curate and we have organized is bulk RNA-seq and single cell. What you are discussing in more on the metagenomics realm, where you also have microorganism which is affecting, and you're trying to understand the host pathogen interaction. So that we offer as services, and it's a more of a collaborative project that we analyze together. But in terms of collection of metagenomics data is not currently that we offer. But for sure, we have expertise in it, so we can help you with your analysis.
Yes, ma'am. That's
Shruti,
all.
you want to add something?
no.
All right.
Thanks. Yeah.
Okay, thanks, Tamli. The next question we have is from Ashish. What technique do you use for unsupervised clustering?
So currently we use the Leiden algorithm.
Okay. And is there an option to choose from different classifiers?
Yes, that can be taken as a part of a customized annotation and curation.
Okay.
Just to add to that, is that so what we intend to do with all the public data sets that we have is to follow a standard protocol, follow a standard pipeline. But of course, we understand that the parameters might not be, like default parameters may not work for everything. But at least from the interoperability point of view is that whenever we are doing something at large scale, we want to follow through a standard pipeline and follow it. So our flow, as Shruti said, we use Leiden clustering, but in the past we also use Leuven. And that's something that we can always tweak in the in our workflows.
All right. The next question: do you only perform analysis within individual data sets, or does your integration techniques allow you to perform cross data set, cross study analysis?
Okay, so I'll take that up, and Richa, you can add. So the process that we follow, right? If we start from raw counts, we are actually, as Richa said, right, we are trying to keep the pipeline as standard as possible. So this does help in doing a cross study analysis. So we do have a couple of examples where in from geo data sets, right, different GSE IDs which were for similar disease, like same disease, doing cell annotations for similar kind of cell clusters. We were able to integrate a couple of data sets from different studies altogether. The most extreme example that I can remember is like we have a data set which is like one analysis, and the data is coming from two different GSE IDs, one EMTEB ID, and one smaller publication. This was something related to COVID and healthy cells, but those four different data sets or publications we were able to integrate owing to the standardized procedures that we follow. So again, we took it from the raw counts. We did not take any information from individual publications, but we standardized it across from all of the studies, and we could integrate it into a one analysis. So that's doable, but with a caveat, we should have rest of the parameters in place to actually do those integrations.
Okay.
Yeah, Richa, you can add.
Yeah, I think you summarized well.
Okay, great. How can we perfectly annotate the subcell types, for example, TH1, TH2, TH10, etc.? Is there a facility in this portal?
I'll take a shot at it again. It's most basically like the main point is trying to find those individual markers that can actually differentiate those TH1, TH2, and TH10 cells. And we are doing some pilot wherein we are going with no information altogether, and we are trying to annotate the cells even to a subcellular or subtype level, but I can't comment on the perfectness of it as of now. Richa, yeah, you can add.
Yes, I mean, I think the main thing is that do we have marker genes available for these cell subtypes? And as you in one of the slides you explained, right, like we do a clustering based on the broader cell levels, and once we have that cluster, we can then do the subclustering and so on. So in that aspect, we can do it better in that way, but it completely depends if we know the markers. And in terms of doing it on the portal, it's currently like a value add services. If you want further curation, that's something that we do in-house with our experts curator. But then the data set can be made available with custom curation as well.
Okay. Thank you for the presentation. Your database looks very impressive. I'm a bit curious about the number of cell types reported in your slide, around 900. Are the cell type labels homogenized across different data sets?
Okay, so yeah, I mean, intuitively this number sounds very high, but when we talk about different levels of uh, of cell types and annotations, right? I mean, even the standardized ontologies have like your broader maybe myeloid lineage, then the T cell, lymphoid lineage, then the T cell, B cells, and these. So it's kind of a hierarchical tree, which is like adding up to the nomenclature, right? The length of the nomenclature, as I would say. So that's where those are different 900 cell types. They can be child and parent relationships.
Okay.
But they are homogenized across different cells, so different data sets. So if it's a B cell will be B cell across all the data sets if it is not being globalized or detailed out in a more finer way.
Just to add to that, so what you will not find on the platform is someone calling P hyphen cell, someone calling it B space cells, or capitalize and so on. Those that's not what this number 900 would represent. This number would represent the way granularity of which we have annotated cell type for different data sets. And the other thing that it represents is that we kind of are using again using what the authors have provided. So we could have cell type for both the level and then also the subtype level that would be included in these 900 ones.
All right, great. We're a little over, but that's okay. Just I think one last comment or question you might be able to. So, I mean, I think what Elucidate is providing the industry is incredible. There are a lot of people behind this. And I'm assuming as you're learning and doing things, you're trying to figure out how to automate things as much as possible. So that would be kind of like a statement question. And imagine providing this to clients. It's got to be a significant cost savings because there's several reasons for that. How are you validating, the accuracy and some of this work? Are you putting some of those constraints in place to validate along the way, et cetera?
Right, so I'll take this. So, but two things, like one of the ways, so currently the process of our cell type annotation is through manual curators and using marker gene that we're using. Coming up with the automated methods, again, we have explored a couple of automated methods, with different types of algorithms that are there, either reference-based or correlation-based. But those are not, those are upcoming ones that we would be doing in automated fashion and be providing with each data set. Yeah, in terms of how we can validate, the first thing is if we are doing it from manual curation point of view, the validation is our publication. So the reference, the publication itself, and trying to replicate those figures that we see is one of the ways to validate. The other information, I think, is as we see very multimodal single cell analysis that are happening. So many times there is site-seq data where you also have certain marker genes which are also expressed, and even those information, even spatial transcriptomics, so they also provide. So all of the information within the publication that could help us in more accurately identifying is something also we use more often in our curation process.
Okay. And then, of course, I think journals and papers and submissions are getting better and better over time, but there's still probably publications out there that are missing key information like, annotation and other things like that. How are you including or excluding, some of that work or that data to, maybe there are nuggets in there that you could take from it. So how are basically you dealing with missing annotation or missing information, et cetera?
I'll take a stab at it first, and maybe then you can continue. So, John, what we're trying is like this is an unsolved problem, and we are piloting a couple of ideas onto it. So, one of the idea is where we have absolutely no information available, right? Not the cell type annotations, not the markers. What we're trying to do is we are trying to use the automated method, and not just one. As we see in literature, there are a lot of cell type annotation methods available. We're trying to get an ensemble of it and try to see the accuracy of it on currently known cell type datasets. And then we are also trying to reinforce or feed in additional information that we are curating from our manual processes, which goes to an approximately 80 plus accuracy. And that kind of boosts the accuracy of the data sets with no information available to around 60%. But then this is again a pilot and a trial thing that we're doing right now. But yeah, we're definitely trying to solve that problem parallelly.
Okay. Great. No, this has been extremely informative. I hope the audience got what I got out of it, but I'm seeing a huge service here to the industry, and it's just going to lead to that model quality data that I brought up in the beginning, where people are going to get more insights, more discovery. I like to say get it done a lot faster. So instead of something taking 4 years, it might take 2 years, right? Or 50% of the time. That's huge for patients out there that are looking for some sort of relief or treat, prevent, cure type of approach to things. So great job. Looking forward to more. And thanks everybody for attending. And of course, if you have any more questions, you can reach out to Elucidata directly. This will be recorded and you can go back and watch at your heart's desire. So thanks, Richa. Thanks, Shruti. Appreciate it very much. Great job.
Thank you.
All right. Take care.
Thank you.
Bye,
Thanks,
everyone.
ma'am.
Thank you.
Bye.
Bye-bye.